I'd like to start talking a little bit about the viral epidemics of the last decades. The first SARS-CoV that we had was in 2002, over 29 countries in East Asia. Then we had H1N1, that is an influenza A subtype in Mexico and United States, and then became a pandemic. This was between 2009 to 2019, and we had outbreaks in India, the Maldives and Myanmar, Pakistan, Malta, Morocco, and Iran. Next, we had MERS-CoV in 2012 that started in Saudi Arabia, and this virus has a 35% death rate. And we had deaths every year from 2012 to 2019, but we have severe cases in the USA and Netherlands, Philippines, South Korea, United Kingdom, Kenya, and over 26 countries. Now we are living through SARS-CoV-2. There is a pandemic that started in 2019, and as of February 2nd, we had more than 376.5 million confirmed cases, with 5.7 million deaths worldwide. And as you know, we've been through Alpha, Beta, Gamma, and now we are dealing with Delta and Omicron variants. So this will not be our last pandemic, and this just highlights the importance of developing tests that can be deployed in specific situations, so we have data to make decisions that best safeguard public health care. There are different types of testing. The molecular testing looks for genetic material of the virus in an active infection, and they are PCR-based methods. So COVID doesn't have a DNA, so we have to do a reverse transcriptase step in which we convert RNA to DNA, and then this DNA can be multiplied a billion times, and then we can use a fluorescence tag to detect the virus. There are the serological tests that detect past infections, detecting antibodies produced by individuals, and the gold standard is sandwich ELISA, in which I have an antibody against COVID, in this case in a well, then you add your blood, and then you can add a secondary antibody with an enzyme, and then you add the substrate that changes color, and then you relate this concentration with the antibody produced by these individuals. The third type of test is the antigen test that looks for pieces of proteins in an active infection, and this is the type of assay that I'm developing as a point-of-care assay. As you can see, you have SARS-CoV structure here, and I am measuring the spike protein, the S1, S2 subunit of this protein. I am using localized surface plasma resonance. Basically, when you have metallic nanoparticles on a surface and you change the refractive index of the medium, this changes the extinction spectrum that you measure from that. So basically I have plastic strips covered with silver nanoparticles that also have the antibody on top of it. And then when I add this to this sick patient's saliva, for the patient's saliva, if they are healthy, there are no change in this extinction spectra, and if they are sick, there will have a red shift of the maximum of this spectra, and then I can relate this to the concentration of virus in the saliva of the person. So the idea here is to construct a simple, cost-effective, and easily scalable immunoassay for rapid SARS-CoV-2 detection. After doing some optimization, I got to a point in which I have maximum extinction shift that is higher for higher concentrations of antigen, which is good. But I was also having a very high background of the saliva alone, and I was using influenza A as a control, and I was having shifts of the same magnitude for both influenza A and COVID. Then I had to optimize even further. So here I'm showing the optimization since the beginning. So for film preparation, if you have different types of nanoparticles with different shapes, you can achieve different sensitivities of these films. I try to do cubic and rod-shaped nanoparticles, but they degrade easily when I add them to the attachment promoter to the plastic. Then triangular nanoparticles, they have a very good sensitivity, but when I was able to make good films of them, but then when I was doing the next steps, it was degrading. So I had just two candidates that worked well. Both are spherical. One is gold nanoparticles and the other one silver nanoparticles. And between them, silver nanoparticles had the better nanometer per refractive index units. So as you can see here, I have my plates, and they were very stable for months, and they had very little variability well to well, which is good. And then I had to work on the absorption or conjugate of the antibodies to these films. And I tried the same way as we do with ELISA, that is traditional with carbonate buffer, and also self-assemble monolayers. Both of them worked, but self-assemble monolayers worked a little better. Then antibody range, I tested ranges that I could put in my strips. So basically, the more antibody that I have, the better signal I have, but these are very expensive. So I was trying to find a point in which I have a good response, but I wouldn't break the bank. And then I have to neutralize the virus in the last step, but I cannot degrade my sensors doing this. So I have to work on this virus neutralization without interfering with the assay. Then I have to try saliva to see if I could just dip this into saliva untreated, or if I have to dilute or centrifuge, and also which volumes I need to use. And lastly, the blocking part. So I block chemically because I use self-assemble monolayers, so ethylenamide blocks unreacted sites. And then for physical blocking, I tried PVP 10, 29, 40, 55, PVA, fish gelatin, BSA 2020 in various concentrations. And I was also mixing and matching this. But in the end of the day, I was still getting a high background of the saliva, and I was still getting cross-reactivity with influenza A. So after talking to people and thinking about it, I hypothesized that I was still having some of my films unreacted. And then I decided to work with DMSO, the molecule that you can see in your left. And basically, this is a small molecule in which the sulfur part reacts with the silver, covering unreacted parts of my film. And after playing around with all of this, I got to a point in which I do have a linear relationship with the concentration of antigen that is spiking my saliva with the extinction maxima shift. And as you can see, the saliva background got very low, and also the cross-reactivity with influenza A. The sensitivity of this test is 15.5 micrograms per ml of the viral S1, S2 subunit. And also, I was able to use only 15 microliters of saliva, which is good. Next steps here, I send this test to University of Montreal Hospital Research Center, and they are using with patients' saliva that are sick and healthy. And they're gonna send me back these, and I'm gonna measure, and then I'm gonna correlate with PCR for viral loads that they will provide. And then I can see if this works in a real setting, because it's very hard to relate viral loads with concentrations. So then I can see if my test actually works. And I am at the same time working in the sensitivity improvement. I am working on that film that worked very well and had a high sensitivity, but it was not very stable. So I'm working in this at the same time on the sensitivity improvement. So summarizing here, there are a variety of tests that can be that will be necessary for SARS-CoV-2 and also the next pandemics that will unfortunately happen. And here I'm developing a test that is very, very cheap. This is the advantage of this test, and uses also saliva instead of nasopharyngeal swab or blood, and it is quantitative to a certain extent. So this would be a good test to deploy in busy centers as a screening test because it's very cheap and easy to use. If my shift was very, very big, I could see this change in color of the strip. But since as of now, the shift is not great, it's not very, very large, we cannot see it by eye. So we are developing a cell phone app in which you can take a photo of the strip, and then the reader will tell you if you need further testing, if you are sick, or if you are healthy. And thank you for listening to me. This is my group. This is the photo that we took in the Christmas party. And I'm here. Thank you so much. And please ask your questions.
Okay, well, I've got a question. It has to do with something you touched on right at the end there. You mentioned that the shift is quite small for your device in spite of the significant shift per refractive index unit. So could you maybe just elaborate on that a little bit?
Yes.
Yeah.
Let me share it again. This is due to how this works. So this technology, LSPR, this exponentially changes where your nanoparticle is. So you have a film of nanoparticles here, and if you have something that interacts very, very close to the surface, you have a very high shift. And if you put antibody here and then you have your antigen on top, it's farther away from the surface. So being farther away from the surface, you have a smaller signal, and that's the nature of this technique. That's why I was able to improve the saliva control, just blocking the surface, because the problem is, even if very little of the surface was exposed, these would have a very high signal compared to what was farther away from the surface. So this is an exponential relationship. So that's why I have a very great shift per unit index when I measure the film alone, but this decreases exponentially when I top up with antibody and then with the virus.
Okay, great. Thanks.
Yeah.
That it's very clear. I see Leah has raised her hand, so.
Thank you, Wesley, and thank you, Ari, for a great presentation. I was curious, would this test work for other variants of COVID?
Yes. So this test can virtually work with anything that you want, as long as you change the antibody that you put there. These ones we tested only with the Delta variant so far, but this depends on how well the antibody works against these variants. So I haven't tested with Omicron, so I don't know if the antibodies that I'm using would work well with Omicron or not. But you can basically just change the antibody that you use, and then you can work with different variants or even different diseases. I even have here, I tested using influenza A antibody against influenza, and we had the results also worked well.
Great, thank you.
Now we have time for just one more question, and I see that we have one in the chat. So the question is, are there any specific storage requirements, specifically in terms of temperature, for this detection kit?
No, the only requirement is that the strip has to be moist at all times, because it can be dried for small periods of time, but if you try to let it dry for long periods, the films collapse. So basically, the way I would do it is just have the strip in a small container that has PBS or something that just keeps it moist, and then you can just dip in the saliva of the person and read it. So I think after 35 degrees Celsius, this wouldn't be good. So there is some temperature control that has to be made, but it's just if you have it very hot, like 40 degrees Celsius, then is a problem. But as a rule of thumb, no, doesn't have much problem with temperature.